Number's still ticking up. I will burn a 30 seconds by saying welcome to the virtual conference room of the Center for Global Discovery and Conservation Science. We are a growing, new but really rapidly growing center. For those of you who are joining who don't know us too well across the ASU campus, we have just hired our 8th faculty member to join us. So we have 8 core faculty and we're growing labs. We're based in Tempe, currently on Biodesign C and moving to ISTB 7. And we're also based in Hilo at our facility, co-located with the US government. So you can find us in both locations. And part of our goal has been to start to broadcast that we're here and to use seminar series as one way to do that. So this year we decided to start a seminar series in coastal marine science and management. And I'm really happy that Dr. Shauna Fu has put this seminar series together, and I'm going to turn it over to her to introduce today's speaker. Shauna?
Hi, everyone. Thanks again for joining today. So I'd like to introduce our speaker, Dr. Jonathan Whitney, who is an ecologist and geneticist in the Ecosystem Sciences Division of NOAA's Pacific Islands Fishery Center in Honolulu. So Dr. Whitney earned his Ph.D. in ecology, evolution, and conservation biology from the University of Hawaii at Manoa, where he combined population genomics, community ecology, and behavioral experiments to explore the origin and maintenance of coral reef fish biodiversity. Completing a joint postdoctoral research fellowship with NOAA and the University of Hawaii, he investigated the biophysical interactions of larval fish and surface slicks in Hawaii, which will be the topic of the seminar today. So we will have time for questions at the end, if you could please hold them until then. With that, it is my pleasure to welcome Dr. Jonathan Whitney with his talk, Hidden World Just Below the Surface. Biophysical coupling in ocean features creates pelagic nurseries with ecosystem scale impacts in Hawaii.
Thanks, Shauna. Thanks, Greg. Yeah, thanks for the invitation. It's great to connect with my Arizona people and build these Arizona-Hawaii connections. It's also a great excuse for me to put on a different shirt, which I really appreciate. My wife Allison got me this for my birthday, new Fish Aloha shirt, so first time I sported it. Still wearing sweatpants, but at least, you know, it's different. And yeah, I'm real excited to connect with my Arizona peoples. I haven't, even though this is virtual, I haven't given a talk in Arizona since my baccalaureate 15 years ago at Prescott College. So this is kind of feels like a return home, even if only virtual. But today I want to tell you guys about a special place in Hawaii. A very cool place that I'm very excited to tell you about. A place where all of these animals, larval stages of more than 100 species of fishes from different ecosystems, ranging from shallow coral reefs down to the bottom of the deep sea, all aggregate in the same nursery habitat for the first few weeks of their lives, where they find these rich hotspots of productivity and really these interconnected highways for larvae. And it all happens because of the biophysical coupling with these dynamic ocean features called surface slicks, which we call bioslick. But first, I want to try to shrink ourselves down to the perspective of an egg. So life for most marine fishes starts with the hatching of an egg. And to survive in the open ocean, these tiny little larvae must find food, avoid predators, and navigate ocean currents to find their way back to whatever their adult habitat is. And the fate of larvae during this stage will have crazy implications for the replenishment of fish populations that we rely on. And in much of the tropical Pacific and in many places, we actually know very little about what happens during these really amazing odysseys. And this is what is called the black box of larval ecology. Basically, after eggs are spawned, we know very little about what happens before, say, a reef fish makes it back to the reef. We know very little about where larvae go, what habitats they use, and how their ecology and behavior shape their populations. And this lack of knowledge really makes it hard to predict things like larval dispersal and recruitment, which are huge for stock assessments, and really for understanding how future changes will affect these animals and the ecosystems that they're a big part of. So a major goal of mine at the beginning of this project was to really try to find where larvae go, and specifically how they interact with dynamic ocean features that can tell us about their ecology. Which brings us to slicks. Now, the people of Hawaii have long recognized that slicks are an important part of the seascape. And when you look out in the Kona coast of the Big Island, they're really hard to miss. And they've even been referenced in old Hawaiian meles as Kekai Ma Okioki, or the Streaked Sea in the peaceful seas of Kona. And despite this historical knowledge that hinted at these features being important habitats for fish, what's in them has really remained hidden to science until now. But let's take a quick step back to orient us. So here we are in the main Hawaiian Islands, a chain of high volcanic islands characterized by really steep bathymetry and deep channels between the islands that are exposed to relatively consistent northeast trade winds. And then specifically this area here in West Hawaii in the lee of the Big Island is where we do most of our work. And this is the core domain of our region's integrated ecosystem assessment program. So this is NOAA's big push to implement ecosystem-based management. And this is our region in Hawaii, the West Hawaii region, which our group led by my colleague, Jamie Gove, aims to provide ecosystem science to support the effective ecosystem-based management in this region. And it is a very special place. West Hawaii is home to the longest contiguous coral reef in the main Hawaiian Islands, which supports a crazy abundance of tropical reef fishes and corals with really high endemism rates. And then in the pelagic realm, or the offshore open ocean realm, it's highly productive. This is a hot spot for marine mammal diversity. You can see nearly 20 species of Adonicetes all within the same day, potentially, if you're really lucky, which doesn't happen, but they're all there. Huge populations of resident false killer whales, pilot whales, lots of other toothed whales. There's a big pelagic game fishery, a sport fishery for tuna and billfishes. I've seen schools of more than 2,000 hammerheads. And there's really just very productive offshore hotspots. And so it's really a special and unique place. And one reason for that is that because of the steep bathymetry or island topography, we get a compression of the coral reef, the offshore pelagic, and the deep habitats in really close proximity to the island. So for example, this area down here along South Kona, within 1 kilometer from shore, you can be in depths of more than 1,000 meters. So it's really steep, basically following that steep volcanic coastline all the way down. So you get these multiple ecosystems really kind of jammed together, and it creates this pretty special effect. Also, the leeward side of the Big Island here, it's in the lee of these two massive volcanoes, 4,000 meter plus shield volcanoes, which do a really good job at blocking the northeast trade wind. So we see a very strong lee in the lee of the shield volcanoes, which creates warm, stratified, and relatively calm waters throughout the year, which are perfect conditions for bioslick. And so this is a synthetic aperture radar that helps to make that point that there's a big lee behind those big volcanoes. So this is synthetic aperture radar from the Sentinel-1 satellite. White areas are relatively rougher, black areas are relatively smooth. And so we can see the wind wrapping around the island to the north and south, and then in the lee, relatively calm, protected waters. And then if we zoom in there on that red box, you can see individual surface slicks. So what are slicks? So slicks are these smooth meandering lines of convergent flow, which is currents coming together at the surface. And they're common in coastal areas around the world. And who here has seen a slick? Raise your hand. All right, I'm pretending that everyone's raising their hand, at least most of my marine people, as you can see them in most places. Sometimes they're called debris lines or foam lines. And for my Gulf California people that might be here, this is a shot, synthetic aperture radar Baja around East Ankel, showing series of internal wave slicks propagating along the coastline, and some of which can be seen here off of San Esteban. So these are seen all over the world. They're kind of common and ubiquitous. And my mentor, Bruce Mundy, just recently put me up or showed me this quote. This is from Ernst Haeckel back in 1890, where he called these features zoo currents or planktonic streams. And he noted that they were among the most noteworthy and important phenomenon of marine biology and needed to be studied. And they've gone nearly a century without having very much attention. Now, slicks are what we're calling bioslicks to differentiate things from oil slicks, but these bioslicks are generated from both physics and biology. And they're convergent zones that are generated by multiple different mechanisms, primarily internal waves, but also headland fronts can make them, tidal mixing fronts, and even submarine groundwater discharge fronts from their plumes, as well as Langmuir cells when you get high enough winds can make those streaks at the surface. So multiple generating mechanisms, and we can, if anyone's interested, we can talk more about it during questions if they have more particular questions about the physics. I won't go into it in too much detail. But we know from past work on internal wave slicks, most notably by Alan Shanks in the US and Mike Kingsford in New Zealand and Australia, that the convergent flow, the surface currents coming together, basically creates a downwelling current that can accumulate buoyant and upward swimming animals right at the surface. And this can impact larval transport and even settlement as they aggregate larval stages. Now, most of what we know comes from temperate, nearshore, shallow shelf systems, which characterize a lot of places, but we don't know the ecological impacts of slicks in the tropics or near islands like Hawaii, where they're a major part of the seascape year-round. And this time lapse helps to highlight that, where this is a normal day in South Kona, seeing slicks form and unforming, moving and propagating along shore. They're really dynamic. They're really important and dynamic part of the coastline. So what are the ecological impacts of these features where they dominate the seascape? And that's what led us to start this project. So starting in 2016, we surveyed most of the west coast of Hawaii Island using a lot of new sontos, some mostly from small boats, but also from the Oscar Elton Celti, the big ships, using surface IK tows. And we did tows inside slicks and also in ambient water nearby slicks as a baseline for comparison. And all of our tows were within 7 kilometers of shore and in bottom depths between 100 and 2,000 meters. So a big range. And we kind of quantified everything that's at the surface, including right above the surface, down to about 1 meter. We counted everything in there pretty comprehensively, everything from debris, zooplankton, on up to fish. And this is coupled with a lot of other data with acoustics, BTD and current profiles, nutrients and chlorophyll, which I won't get into all of today, but part of a much broader project. So here are the first broad strokes of what we found. We're going to see several plots like these that show the ratio of what we found in slicks to what is found in ambient water, along with 95% bootstrap intervals. And so on average, we find nearly twice as much phytoplankton, 4 times as much zooplankton, and about 8 times as much larval fish on average in these features. So it's kind of almost a doubling from each trophic group going up, starting with phytoplankton. So let's first dig into this and explore the invert zooplankton community first. So slicks accumulate not just dense, but really diverse zooplankton. And this plot is the same as before, shows the ratio of mean densities in slicks to ambient water with the bootstrap intervals. And then the dashed line at 1 indicates equal abundance. So something right on along that line would be just as abundant in versus out of slicks. And so amazingly, 80% of the holozooplankton groups, so things that aren't larvae, are between 2 and 25 times more abundant in slicks. So this is everything from copepods and other microcrustaceans, even terrestrial insects and water striders, which we'll talk a little bit about later. So really, other than heteropods, the entire surface community is compressed into these dense bands. And we also get lots of larval invertebrates, things like gastropod veligers, crabs, shrimp, and somatopod larvae, more than 5 times more abundant in these habitats. And collectively, many of these groups are prey for larval fish, which we found an incredible diversity of fishes that accumulate here. So Slicks hosts at least 112 species of fishes from 54 different families, incredibly diverse. And these are a mix of coral reef fishes, lots of different species of jacks, damsels, triggers, barracuda, and then we have epipelagic fishes, so fishes that are living offshore and at the surface. And these are things like flying fish, mahi-mahi, and billfish. And interestingly, even deep water fishes like anglerfish and lanternfish, all interacting in the same hotspot for these couple first weeks of their lives, within only a few kilometers of shore. And the deep connection is particularly fascinating to me. So, for example, we get lots of larval tripod fish at the surface. Their adults are living at the bottom, somewhere between 2,000 and 4,000 meters. And then their larvae are floating up to the surface as in the egg stage, hatching, and for the first few weeks of their lives, chilling at the surface, eating and being beautiful. So these guys, we believe, are mimics on siphonophores, at least that's the hypothesis, and I buy it. So at the surface. Another really cool example of this is with these blind cusk eels. So that the adults are blind. They have these lost recessed eyes in the adult stage that are basically just living in complete darkness, 4,000 meters at the bottom of the ocean. Their eggs are floating up to the surface, and these little larvae, which have big eyes and are visual predators for the first month of their lives or so, living and feeding at the surface before then descending back down 4,000 meters. Think about that pressure difference when you're 1 inch long, and then losing their eyes, becoming blind, and living at the bottom of the sea for the rest of their lives. So really incredible life histories that we get here at the surface. And so we get larval and juvenile stages of some fishes that you might recognize, like these big game fishes, the mahi-mahi up top, the short-billed spearfish in the middle, and the giant alua on the bottom, as well as coral reef fishes, like these puffers, or porcupine fish rather, and even several species of deep sea anglerfish, like the humphead. So incredible diversity that we get. But if you were to pick 8 fish out of a hat from all of our samples, the bulk would be these guys. So these 5 families represent more than 80% of all the individuals that we collect. So these are like the main players here: flying fish, chubs, half-beaks, damsels, and goatfish. And they share some similar morphology in that they're all really melanized. They have a lot of dark pigment. And we believe that that's an adaptation to handle the higher UV stress from living right at the surface. Okay, so that's the diversity. Now let's explore abundance. So this is data from 33 of the most common common families, fish families, that we find in these habitats. And this is the same plot again, essentially showing the ratio of slicks, what we find in slicks to ambient water, and that dashed line equals an abundance, equal abundance. So 88% of all fish families in this category are at least twice as abundant on average, and that's a mix of reef fish, epipelagic, and deep water fishes. And then these over here are somewhere between 10 and 100 times more abundant. So really pretty much only found in these habitats. And we were really surprised to find that so many species, even entire families, and in this case, the entire order of Balaniformes, the flying fish, needlefish, and half-beaks, they only occur in these habitats, which suggests that they're dependent on these critical places during their early life stages, which we did not know before. And one of my favorites of species that we only find in slicks are these man-o-war fish, which aggregate in and around and even feed on the Portuguese man-o-war. They're really cool little fish, and then as adults they go deeper, but as juveniles and larvae they're right at the surface and are specialists in the slick habitat. Okay, so taking a quick breath. So we find lots of things there. We find diverse and abundant aggregations of animals, but really what are the ecological impacts to individuals, and then thinking ultimately ecosystems. So from what we found in the water, we needed to try to scale up our in-water surveys and combine them with a new method for detecting and mapping slips. How can we scale up what we're seeing in the water to the more regional ecosystem level? And here's where the collaboration with Greg has been instrumental, and with GDCS. So when we posed this problem with Greg, he and Jiwei figured out how to use the Dove constellation imagery. I'm sure that many of you are working with the Allen Coral Atlas. And really, this was kind of the layer, the surface layer that was getting thrown out to look at coral reefs that we were really interested in highlighting to see if they could figure out how to classify slicks, which they did figure out. And for the first time, we'll be able to map slicks at scale and isolate them from satellite images, which really highlights how ubiquitous they are up and down the coastline. And they had never been mapped in this way before with this resolution, and are really helping to show how important these are at an ecosystem level. So using the slick mapping areas, we scaled our in-water surveys to this 1,000 kilometer squared study area. That's the dark blue shaded region there, basically 7 kilometers across the whole West Hawaii coastline. And although slicks only covered about 8% of the ocean surface, they had disproportionately dense composition, having 39% of all the surface dwelling larval fish, more than 25% of the nusonic zooplankton, and 75% of all the floating organic debris. So things like leaves, sticks, and algae. So really, it's a large proportion of all these larvae, along with the resources, food, and shelter that they need to survive, which tells us that they have a critical influence on the larval ecology of this region. Now, it's important to note here that we're really characterizing and talking about what's at the surface. And this drifter data helps show how slicks are really surface features. So this is data from an acoustic drifter that we basically let a slick pass by. And so you have the estimated backscatter of particles in the water, which is phytoplankton, zooplankton, and fish. Yellow is more and blue is less. And this kind of shows a cross section of what we see from backscatter going across the slick. And these are two different slicks that we're showing data for. And what this shows is that they really impact the top 5 meters, which is the main point here. So they enhance what's in the water column in the top 5 meters. So they're a very surface feature. And so that's important to keep in mind when we talk about these percentages here, that we're really characterizing what's at the surface. There's a lot of other species. In fact, most fish species are distributed, the larvae are distributed deeper in the water column. But these features are incredibly impactful for around the 10% of Hawaiian fish species that are nusonic or that are living right at the surface. So for this surface community, what are the ecological impacts? So we put this conceptual diagram together to try to visualize the wealth of ecological connections enhanced here. So starting at the base of the food chain, we see that slicks aggregate phytoplankton, zooplankton, eggs, larval invertebrates, and larval fish, and even insects right at the surface through a combination of both convergent flow, which is a surface current coming together, and end behavior, which I'll get to in a minute, which leads to greater spatial overlap between larval fish predators and their prey, and creating more dense resources. It increased encounter rates, we believe, create better foraging, faster growth rates, and then presumably higher survival rates. So that larvae that are in slicks are then radiating out and replenishing adult populations in multiple overlapping ecosystems. So we have fish going to coral reefs, fish going to the bottom of the deep sea, and fish staying out in the epipelagic. And we see multiple different functional groups. For example, say coral reefs, we have larvae of top predators, grazers and browsers, planktivores, and cryptic macrobenthics like gobies and blennies, as well as the macrocrustaceans. So lots of different functional groups that are then enhancing those coral reef ecosystems. And then when we add in all of the trophic links, really start to see how important these connections are for these multiple ecosystems. So all the blue things are then going up to the things that eat them. So enhancement of larvae, for example, flying fish, which we found almost exclusively in slicks, are then huge prey items for lots of different pelagic predators. Flying fish are eaten by almost all Hawaiian seabirds. They're eaten by a bunch of top predators like mahi and billfish, which also live in slicks as larval and juvenile stages, and they're fed on by marine mammals. And then those top predators, many of which are then eaten by us as humans, particularly the game fish. And then even on the coral reefs, a lot of the animals are either directly or indirectly fed on by humans, like the big giant alua, which are found condensed in slicks as well, are a highly prized target for recreational fishers. So really all of this together, we hypothesized that the increased predator-prey overlap in all of these enhanced ecological connections are really amplifying the flow of energy up the food web, up to top predators, even up to humans, which should ultimately enhance ecosystem productivity in these multiple ecosystems and even fisheries productivity. And one really cool unexpected thing that I wanted to share is that we found that terrestrial insects were really highly concentrated in slicks. This was unexpected. So things like bees, bugs, moths, beetles were 26 times more concentrated in slicks than in ambient water. So there's these land insects getting blown out to sea, fall on the surface, and then get compressed here. And then they're fed on by these water striders, which are Halibates. They're the only genus of marine insects, and they're really adept sea skaters and to be out at sea. And we failed to find a slick without these guys already skating inside them. So they're kind of like the first responders as slicks form. They show up and they're filter feeders on dead insects. So their prey are highly concentrated there. The water striders themselves are eaten by seabirds, even a species of shearwater that feeds on them exclusively. And then they're also fed on by larval and juvenile stages that are accumulating in slicks. So you get enhancement in multiple different dimensions, all in these compressed areas right at the surface. So in short, lots of ecological connections are being enhanced here. And one of the questions was, is this due to just physics, or is this also behavior? And the short answer is, we believe it's both. So we can assume that eggs and tiny hatchlings that aren't very good swimmers are passively accumulating, just like debris does. But we find evidence that suggests that late larval and juvenile fishes are actively selecting slicks. Excuse me. So we measured more than 13,000 individual little fish. And when we look at the size structure, the frequency on the y-axis versus the larval length on the x-axis, we see a couple of things. First, there are more smaller individuals and less large ones, which is expected given their really high mortality rates. Roughly speaking, of all the eggs that hatch, probably 99% of them will die and only a few will survive. So we expect to see this size structure, more little ones, less larger ones, all of which are more abundant in slicks. But particularly, all of the larger individuals, greater than 2 centimeters or so, are pretty much exclusively found in slicks, things like this larger juvenile mahi-mahi. And when we model this relationship, we find that the probability of occurring in slicks increases with larval fish size. And what also increases with size is they become more competent swimmers. So essentially by these larger sizes, like this pair of juvenile filefish, they're basically micronecton. They're good, strong swimmers. And so we find this to be compelling evidence that these larger fishes are actively selecting this habitat to capitalize on the concentrated prey and also the shelter. So in a world without structure, floating debris really provides shelter from predators. And we know from adult stages that adult fish are attracted to floating debris. And this is why FADs, fish aggregation devices, are put out. And we find good evidence that debris is a really strong predictor of fish occurrence in flicks as well, in larval and juvenile stages, and is an important cue, probably a more important cue than prey, to driving or bringing fish into flicks. Now, the convergent flow at the surface, which has a downwelling current and accumulates buoyant things, accumulates all kinds of floating debris, which was measured here using dry weight. And this is organic stuff like macroalgae, plant and animal matter, leaves, twigs, palm fronds, as well as plastics. So we found organics were about 30 times more concentrated here, and plastics more than 250 times more concentrated. So the invasion of hazardous plastic debris represents a real threat. And to try to illustrate that, this is all floating plastic debris from one single tow, a 7-minute tow from Kiholo Bay in 2017, where we found more than 37,000 individual pieces of plastic in a single 7-minute tow at the surface. Now, most of them are really tiny little microfibers and little fragments like the size of sand grains. And to put this into context, these densities are rivaling what we find in the Great Pacific Garbage Patch, in these really compressed areas. And then you can see all the larval fishes all swimming around in this debris with an illustration. So what does this mean? Really, it means that the relationship between fish and plastic has changed in flicks. So in ambient waters, larval fish outnumber plastics about 2 to 1. But in flicks, this relationship changes dramatically, and plastics outnumber larvae by over 7 to 1 and are strongly correlated. So where there's more fish, there's more plastic. So this increases the encounter rates between fish and little plastics. And unfortunately, most of the microplastics that we find are about the same size as larval prey. So somewhere between 0.5 millimeter and 1 millimeter. So the question is, with this strong overlap in size and prey size, do larval fish ingest plastic? And so we tested for ingestion by dissecting the guts of nearly 700 larval fish from 9 families. So fish about 1 centimeter or so. Dissecting those guts is very tricky and time consuming, as you might imagine. And from this exploration, sadly, we found that they do ingest plastic, but not all taxa equally. So these tiny little pieces that are barely visible to the naked eye, like this tiny piece of polystyrene foam, are consumed. But most of them are these little pieces of synthetic microfibers, many of them blue, which we'll talk about in a second. So plastics that you can barely see with the naked eye are what we're finding in the guts of these tiny little fish that are as young as 1 week old. So overall, we found about an 8% incidence of ingestion of microplastics in fish and these larval stages, which is more than double the rate that we found in ambient water. But it's really variable. So the goatfish, for example, we didn't find any plastics in. The mahi were pretty close to that. And the worst was the highest incidence we found in flying fish. So highlights, there's variability, but sadly really consistent among many different families that ingestion seems to be somewhat common. Now, why are fish ingesting microplastics? So we think, like this little piece of microfiber here, are getting confused for prey items. One primary one could be Oicopleura, which is an apodicularian or a pelagic tunicate, which are a major prey item of many different kinds of larval fish and have this kind of long string-like structure. And the blue connection is really interesting because most of the nusanic animals that live right at the surface are camouflaged by being this deep blue and then blending in with the deep blue water below it. So we think that the higher ingestion of blue plastics is probably correlated with the color of their prey. And this blue connection is seen in a lot of different microplastic papers. You see it noted somewhere like, oh, we found most of the plastics happen to be blue, and this is relatively consistent. So there's a lot of potentially impacting health and survival issues with larvae and plastics, because this is a very vulnerable life stage where their bodies aren't even fully developed yet. Organs are still building, immune systems are still building, so they are particularly vulnerable to toxins and any kind of foreign threat. So it's a real threat to the biodiversity hosted in these nurseries. Okay, let's back away from the plastics and let's move back to some more of the more uplifting ecological implications. So one of our next big challenges is to try to incorporate what we're seeing, these ecological enhancements, into ecosystem models where we can really quantify the impact they have on ecosystems and fisheries productivity. So one of those goals was to try to link up in a more high throughput way what's going on at the surface versus what's going on underneath the water without having to do net surveys. So we experimented last year or a year and a half ago, coupling a time-lapse imaging system. This is a camera that's on the Marine Education and Research Center in Papa Bay, above Greg and Robin's residence. Robin is all too familiar with these systems, and I have lots of amazing photos of her climbing up on the reef or climbing up on the roof to clean the glass on these and maintain them. But these are out. This camera is out taking an image of this coastline once a minute for the last year and a half. And so this experiment, we coupled this with an upward-looking current profiler and ecosounder seen here. And so what this is allowing us to do is to try to couple what's going on at the surface through these surface manifestations with what's going on underneath, and try to learn more about, from a longer standpoint, not just when we're able to be in the field, but can we characterize what's going on throughout the year in different seasons and different tides. And you can see the mooring is right here, this little purple dot, in 600 feet of water, which is insanely deep given how close this is to shore. This Papa Bay is wildly steep, and there's the murk right here. So we can see flicks are forming by a couple different mechanisms, probably first with tidal fronts, and then as a wave of internal waves comes, it slides them all and it pushes them towards shore, and we see that debris and all that life coming and crashing right into the coral reef. And when you're on the reef, when these features come, you can see all the planktivores, the triggerfish and damsels are coming up and feeding on these basically pelagic subsidies of zooplankton that are rolling in. So we get these influx of energy flow from offshore to onshore, replenishing coral reef larvae, but also giving them, the planktivores, more energy. So we see this great coupling with flicks that probably influences larval transport and retention. I'll show that video again, just sped up. Imagine larvae developing offshore, and then when they're ready, they're basically getting this extra push that allows them to find their way back to reef. And we believe that this could be a really important mechanism enhancing self-recruitment and reducing dispersal distances, or basically allowing larvae to stay close to the shore and then allowing them a chance to get back to the reef. Now, there's a lot of questions that we get about what happens with pelagic stages that don't want to be near a coral reef. Coral reef is not where a larval mahi-mahi wants to be, for example. And we don't yet know how these animals are avoiding the coastline, but they seem to be doing it. And they may be using cues like reef noise, which is an attractant for a coral reef fish larva, but maybe a deterrent for these deeper and more offshore species. But that's still an open area of research. Okay, so just to sum up before we close, these bioslicks, which are generated by convergence of physics and behavior, hence the bio. Bioslicks are really concentrating larvae as well as their prey, shelter, and predators into these dense structured bands. They provide essentially nursery habitat for coral reef, pelagic, and deep water ecosystems that should enhance larval supply as well as energy flow. They create essential fish habitat and potentially a mechanism for self-recruitment and retention near islands. And we hypothesized that increased predator-prey overlap ultimately leads to enhanced ecosystem and fisheries productivity, which we'll talk about in a second to close. And really that slicks impact the entire surface community with important implications for the replenishment of adults that we rely on. And so the next steps of this are really going to increase our partnership with Greg and the GDCS into a really exciting area where we hope to expand what we're doing to a statewide scale and then potentially more globally after that. So this next goal is to really try to quantify how these features impact ecosystem and fisheries productivity. And in order to do that, we need to get more regional scale metrics to put into end-to-end ecosystem models. So how do slicks enhance phytoplankton across a larger scale to increase ecosystem productivity? And then how does that eventually translate into increased fisheries productivity? And so our next steps are to combine what we've done at a larger scale using the Dove satellite imagery to map slicks across the state, combine that with Greg's, with the GAO's hyperspectral imaging using archived data, and combining that with liquid robotics wave glider to do in situ chlorophyll mapping to really get a more ecosystem level quantification for how biomass is being affected. And then I want to end by taking a further step back and saying that we're showing that these features and the behavioral attractions to them have a big impact in Hawaii, but we do not believe that this is isolated to there. Bioslicks are found in coastal areas all throughout the world, but their ecological impact has really only been assessed in a small handful of places around the planet. And it's possible and very likely that they have important ecological implications in lots of other places, particularly on the leeward sides of islands throughout the Pacific or throughout the tropics, potentially here in the Antilles as well, and lots of other places where maybe all of y'all work. So we are very curious to hear what these features may be doing in areas where you work as well. And with that, I just want to thank our long list of collaborators, but especially Greg and these connections, and G-Way with GDCS that have really turned our work into a whole different direction. And we're really grateful for this partnership, and we're looking forward to expanding more. And I want to give a big shout out to my close colleague, Jamie Gove, who's been a huge part of this project, as well as my postdoc advisor, Margaret McManus at UH, and all of our other collaborators. And a mahalo to all the people that helped in every aspect of this research, and particularly Robin Martin for crawling up on the roof and keeping those cameras operating. So yeah, I guess I'll stop there, and thanks everyone for your attention.
Thank you. That was wonderful and really beautiful photos. So we do have time for questions. If people could raise their hand, I'm going to start off with a question, if that's okay. I was wondering, do these slicks travel in like predictable enough ways that you could incorporate them into marine planning?
Yeah, a lot of them do. So they are formed by a couple different mechanisms, which we talked about. But the internal wave slicks are pretty regular. They're attached to the tidal cycles, but throughout multiple different times of the day, they're coming in predictable waves or sets of waves. And there are some other slicks that are stationary and form in the same places all the time. For example, off of the Kona Airport, Keaholei Point, we mapped slicks 3 days in a row that were in exact same spot, stationary, and these are probably driven by headland fronts or tidal mixing fronts. So yeah, you get many of them are moving in a kind of predictable way, offshore to onshore, parallel to the shoreline, like the internal wave slicks do, and then some of them are stationary. So yeah, potentially they're predictable. They're very dynamic, but potentially predictable enough to work into planning.
Awesome, thanks. JD, did you want to ask your question?
Yeah, this was an inspiring talk. Thank you. I have seen bioslicks at Monterey Bay, where I spent a lot of time, and also from Robin's, Greg's murk as well. So my question is that camera that's on top, I know that you can map this from satellites or from GEO. Have you corrected the perspective to get like a top-down view of the slick from that camera footage? Like, and does it help if you could do that? And how precise will it be? And
yeah, it's a great question. So we've done it on very small scale. We did it for the first camera setup that we did, which had relatively low resolution. And we did it for a few days of imagery where it was geo-rectified. And then we were able to overlap that with observations from the Sentinel-1 synthetic aperture radar data and basically show that they linked up well. So in short, we did that on a small scale. What we're working on now is to turn that year plus of data and do it at a larger scale, which we have not yet done. So that's in the works of basically figuring out how to geo-rectify those hundreds of thousands of images and then really be able to, map their movement, their behavior, and correlate that with the rest of environmental variables. Do we see them only at particular tides? Are they stronger or faster in spring tides versus neap? And all of those real environmental links that we hope to make play when Greg figures out how to do that.
And just to follow up on that, can you retask the wave gliders? Do you do that based on current slick information?
That's the idea, yeah. So the liquid robotics wave glider is cruising up and down the coastline all the time, and we're actually gearing up to do an experiment in the next couple of weeks where we have it run in and out in front of where the time-lapse camera is, and then hopefully be able to correlate and link them up via the images. And
very nice.
yeah.
Cool. Tadia, did you want to ask your question?
Yeah, please. Great talk, Jonathan. Thank you so much. Bioslick, favorite places to hang out in the Gulf of California. Do we know much? Has anybody looked at any of the toxicology stuff, heavy metals or accumulation of fuels and diesel and all this stuff? Like, do we know much about that in slicks?
In short, no. There's anecdotes in the literature that actually there's one paper that showed that oil globules off of Santa Barbara were more common in slicks, and they were tracked there. This is from the offshore rigs off of Santa Barbara. So that's kind of the only thing that I know of that particularly talks about oil. And then toxicology, not that I'm aware of. We did nutrient analysis in and out of slicks and did not find that there was a very big difference in any of the nutrients. And the reason that's pertinent is that we think that there's so much water flow through these features that it probably doesn't trap mass of water very much, but it does trap things that are buoyant, like oil and all the plastic debris and stuff. But yeah, the rest of heavy metals and stuff like that have not been studied, but it's more likely if it's not buoyant that it will get flushed out of the system.
Thank you.
Thank you.
GUA, did you want to go?
Yeah, John, thank you so much for the great talk. That's really excellent. So actually not a question. I'm thinking of because the backscattering coefficient of phytoplankton or zooplankton or particles can be also detected from the satellite images. Maybe thinking of the using remote sensing to measure chlorophyll, also measure some like phytoplankton concentrations in the bioslicks, and maybe also interesting to test in the future. Yeah, just thinking that. Yeah.
Yeah, absolutely. I mean, that's exactly what we're hoping that we can do. Yeah, whatever we can get and characterize the chlorophyll and the phytoplankton biomass and potentially compositions from remote sensing imagery at high enough resolution to pick up what's in and out of the features is really the next big step. So yeah, I'm incredibly hopeful that we can do that with the data product you guys are already working with.
Okay, well, anyone else? Oh, okay, yep.
Can I?
Yep, JD.
Yeah, so that was very fascinating that goldfish does not eat microplastic. I actually recently learned that they have an excellent vision system. So did you mention goldfish, that they don't seem to consume microplastics?
Goatfish.
Yeah.
Yeah. So you learn that goatfish have a more, really?
Yeah, better vision than humans. And I learned this because I was looking at this eyesight of predators at night, like for some research we are doing with AZ Game, and then goldfish popped up, and apparently they have excellent vision. I was wondering if that had something to do with them not consuming microplastics.
Yeah, I mean, that was one of our, we're like, maybe they have better vision than the other fish, but I've never seen any data to support that. That's really interesting. That's great. I would love to see that study. Yeah.
And related to micro, and I'll read up more, and I'll link back with Shauna if I find something.
Yeah.
So the footage that you showed of the plastic, it was very nice. Like it had, has it been automated, the identification, the mapping of that, the counting of different kinds of things in that?
No, it's still incredibly, yeah, it requires a lot of manual labor. Basically what we do, the whole bulk sample, we pick through individually and individually pick each one of the larval fish as well as the plastics out. But then
from the computer
yeah,
side.
And then, yeah, basically we take all those plastics and put them in and digitize them. And then the rest of it is automated, like using ImageJ and stuff like that. But we haven't yet figured out how to do more like a cell cytometry or a high throughput design. And the trick is that we have such a mixed sample. Like if we just had a sample of plastic, that would be easier. But what's in, what comes up from the net is like, it's leaves and it's twigs and it's bees and it's zooplankton and it's fish and it's plastics. And so it's tricky to do like any one more high throughput processing to deal with all of those different groups. So ultimately, like we need to figure out how to do it more high throughput, but this was pretty old school where we manually picked everything, which is.
Yeah, if you could share some data through Shauna, we could chew on that. Yeah, if that's
yeah, okay. Yeah, let's do it. That would be awesome.
Thank you.
Thank you.
Cool. And Tadio, did you want to ask a question?
Yeah, just one last question. It's so exciting. Do you feel like warming ocean temperatures, changing wind patterns, are there significant threats to slicks in many areas? Like,
yeah, I think it's a fantastic question. Yeah, really are. For example, with wind patterns, if wind patterns are shifting in a changing climate, if trades die, how that will affect, these features, huge open question. And particularly with warming, these surface communities, yeah, these animals are really tied to the surface. They are right there, and they do not swim much deeper, at least most of the fish that we're seeing there. And so surface waters would be more, prevalent or more susceptible to warming. And if they get too hot, it's not like they're going to move deeper, that they're very associated with the surface. So I think they're particularly vulnerable to warming.
Okay, well, I think, any more questions? Anyone can speak up? Or if you think of something later, I can connect, Jonathan and you.
Yeah, please reach out.
So thanks again. That was really cool. So please join us again for our next seminar on the 25th of March at 12 p.m. And it will be with Dr. Margot Hein, a consultant for marine ecosystem restoration research and consulting, and she will give a seminar about coral reef restoration. Thanks again.
Thanks, everyone.
Great
to connect with y'all.
Bye-bye. Thanks, John.
Thanks, John.
Thanks, John.
thank you.
All right, thanks y'all so much for hosting.
Thank you. That was so cool. All these photos are just amazing. Makes me miss
Thank
Lolly.
Yeah, right?
Yeah. There's so much to do. I was just thinking, sorry, I'm just asking one more question before I let you.
Please, yeah, go for it.
Like, I wonder